Hi guys, welcome to our channel. This is Science Lectures. Thanks for being with us. Please consider subscribing and comment in the comment section, and you like our videos, please. So we are continuing with our series of the cell organelles, cell biology, cell organelles. So today we want to look at the cell membrane, the outer covering of the cell membrane. So basic structure of the cell membrane. Before we start off, we need to know what the cell membrane is, where the cell membrane is formed. So let's imagine we are having a plant cell, an animal cell, and finally let's say we are having a prokaryote. This prokaryote can be a bacterial cell. So all these cells possess what we call the cell membrane, and it is always interior covering. It is a covering the cytoplasm of the cell. So all these cells possess the cell membrane. However, the cell membrane of the animal cell is not covered by any other layer, so it has no any other layer covering it on top. For the prokaryotic cell, it will of course possess a cell wall, and on top of the cell wall, we said we have what? We have a capsule. Some bacteria will have what we call a capsule, also covering as an extra covering. But again, for the plant cell, we will also possess a cell wall covering the cell membrane. So this is just something to introduce us to the cell membrane of the cell. So it encloses the cytoplasm. It will enclose the cytoplasm, and it will prevent entry of substances in and out as we are going to have a look. So that is very important for us to know that the cell membrane of the prokaryote is always covered with the extracellular wall, and again the cell membrane of the plant cell is also covered by that extracellular wall. Prokaryote and the plant cell. But animal cells will entirely have the cell membrane outside. So let's consider the animal cell. So if this is my animal cell, let's say that is the cell. If this is my animal cell, this outer covering, inside we have that's the fluid, which is the cytoplasm. The outside covering will definitely be the cell membrane. That is my cell membrane. So today we are going to zoom into the cell membrane and see the components of the cell membrane, component by component. So let's imagine that is a sketch of our cell membrane. So this cell membrane will have what we call the phospholipids. We can have a protein. We have a protein that spans along the entire membrane. And this protein that spans along the entire membrane is what we call the integral protein. The integral protein spans along the entire membrane, or we can call it the intrinsic protein. An example again can be the transmembrane protein. So it's a transmembrane protein, spans along the entire membrane. Then it is spanning along that entire membrane. Then we can have another protein which has, like a root 3. This one is also a protein, but it's specifically designed for transport. It's a transport protein. Then we have, let's say we are having our glycoprotein also there, glycoprotein. A protein having a carbohydrate attached to it, the glycoprotein. Then again we have another protein. We can have another lipid. If we have a lipid there attached to the carbohydrate, it's the one we refer to as a glycolipid. We have also a glycolipid there. Then if this is our phospholipid bilayer, this one will be our phosphate head. The phosphate head. Then we have the fatty acids. The fatty acid tail, that those fatty acids are present. So basically this is a structure of the cell membrane. But we are going to look at this in details. We are going to cover this cell membrane in details. So we are going to start with what the cell membrane is. We know the cell membrane, we will define the cell membrane as being semi-permeable. A semi-permeable membrane. Semi-permeable membrane. What does semi-permeable mean? Semi-permeable simply means it will limits entry of substances into the cell, into and out of the cell. Limits entry of substances into and out of the cell. So what is this limiting all about? It can either completely prevent the entry of these substances or exit of substances. Then it can as well limit to some extent. Then it can again allow molecules to freely move along the membrane. So we are going to see molecules that can freely cross via the cell membrane. We are going to see molecules that do not cross the cell membrane, and we will see molecules that cross the cell membrane at a given extent. They have a certain extent to which they cross the cell membrane. So I want us to give the basic components. We are going to see the basic components of this cell membrane, and then the basic components of the cell membrane. So the number one component, they are the membrane proteins. Membrane proteins. We can divide, there are three main components. Then the membrane lipids. Then the third component is the glycocalyx. So those are the main components of the cell membrane. So we are going to first look at the examples of these components. The proteins, we are going to see there are some proteins that are intrinsic or integral proteins that are inside the membrane, but we are going to see there are some other proteins that are peripheral or extrinsic proteins. Then the membrane lipids, we are going to also see these lipids, where we have examples like phospholipids. Then we have cholesterol. Then we have the fatty acids. That are attached there. Then the glycocalyx is divided into two main components of the cell membrane. That is, those are the glycoproteins, and the glycolipids. Glyco comes from sugar. So if we have a carbohydrate attached to a protein, it becomes a glycoprotein. A carbohydrate attached to a lipid, it is the glycolipid. So we are going to start off with the membrane lipids. We are going to have a look at the membrane lipids. So our membrane lipids, we've already seen that this is a phospholipid bilayer. So these are phospholipids bilayer. So it's a bilayer of the phospholipids. But if you're to be so keen, okay, the membrane appears like this, like that. Yeah, like that. So these are the phosphate heads. This is a phosphate head. This is a phosphate head. And we know phosphates are negatively charged. A phosphate head with its negative charge. So the phosphate heads will always point towards the exterior of the cell and again towards the interior of the cell, because that is where the aqueous solution is formed. So the aqueous solution is found outside, either outside the cell or inside the cell. So these fatty acids inside here, that the fatty acids that will hide inside there, for them they are non-polar. So fatty acids are non-polar. So these are our fatty acids, but the phosphate heads are outside, pointing the outside of the cell and the inside of the cell in the aqueous fluid. Why? Because they are negatively charged, and that gives them some degree of polarity. So that is how the cell membrane, the phospholipid bilayer, is simply. That is how simply it is arranged. We have the phosphate heads that are polar. They will point towards the exterior of where the aqueous layer is formed. Then again towards the interior. They will point towards the interior because they are negatively charged. They will attract the water molecules there. They react with the water molecules there. Then the non-polar fatty acids will move away from the water. So why is this concept important? Why is the concept that important? So we shall see the importance of the concept. So we are looking at the phosphate head. This is the phosphate, which is negatively charged. That means it has some degree of polarity. Then the fatty acid is non-polar. So the phosphates are polar. Fatty acids, non-polar. So when transport is occurring along the cell membrane, what will pass through the cell membrane, or what will diffuse via the cell membrane freely? What will diffuse through the cell membrane freely are the small non-polar molecules. What are the examples of these small non-polar molecules? We have oxygen. This molecule is non-polar and very tiny. Then we have carbon dioxide. Also will diffuse via the cell membrane, but with the aid of the fatty acid of the phospholipid bilayer, because these fatty acids are non-polar, and we know like dissolves like. So a non-polar molecule will aid the diffusion, the simple diffusion of another non-polar molecule. That is, the fatty acids will aid the diffusion of small non-polar molecules like oxygen, carbon dioxide. Then we can have some steroid hormones like testosterone, estrogen, such hormones. Then we can have also lipid-soluble drugs. Lipid-soluble drugs will also diffuse along this membrane. So we are looking at this as helping us to form this membrane. The phosphate being negatively charged, some degree of polarity will be found at the exterior of the cell, and again in the interior of the cell, covering the interior of the cell. So literally, if this is our cell, this is what will appear. So these are the phosphate. This is the phosphate head, pointing towards the interior and towards the exterior, because that is where the aqueous layer is formed. So fatty acids being present, they will aid in diffusion, simple diffusion of the small non-polar molecules. Which molecules are small non-polar? Oxygen, carbon dioxide, steroid hormones, and some lipid-soluble drugs will be able to diffuse via this membrane. Why diffuse via the non-polar membrane? Because we know likes will dissolve, like dissolves likes. So that is simply what is all about. We are still looking at the membrane lipids. So we've seen the phospholipids and the fatty acids. But again, fatty acids, we need to elaborate more about these fatty acids. So basically that is what is all about the phosphate. Fatty acid, we said it's non-polar. But again, fatty acid, interestingly enough, these fatty acids, The fatty acids can either be saturated, or they can be unsaturated. Let's imagine this is our phospholipid having a fatty acid attached. So one side, let's say this side is saturated, a saturated fatty acid. This one is unsaturated fatty acid, an unsaturated fatty acid. The unsaturated fatty acid has somewhere, let's say a double bond somewhere in its structure, and the saturated has only single bonds. So presence of the unsaturated fatty acid creates a kink within the structure of the phospholipid, what we call a kink or a bend in the fatty acid structure. So if a kink happens to be in the fatty acid structure, this will aid in what we call fluidity of the cell membrane. So in other words, kinking of the unsaturated fatty acid, I think at an angle of around 30 degrees will bring about the maintenance of fluidity within the cell membrane, as we'll see. So that is the importance of these fatty acids. So fatty acids, saturated, unsaturated, first of all, they aid in the diffusion of small non-polar molecules, like we've seen oxygen and carbon dioxide. Then again, the unsaturated fatty acid will form a kink that will aid in fluidity of the cell membrane. So number one factor affecting fluidity of the cell membrane, fatty acids. If they ask about fluidity, fatty acids, you should not forget to talk about the fatty acids. So which other thing is very important in the phospholipids, in the membrane lipids, membrane lipids. So remember we said the membrane lipids, we said we have the phospholipid, we said we also have the fatty acid. Phospholipid, we've seen the importance of our phosphate group. Then fatty acid, we've seen the importance of the fatty acid, the unsaturated fatty acid. Then finally we have another component which is an important lipid, which is cholesterol. So cholesterol is another important lipid inside our cell membrane. What is the importance of cholesterol? Cholesterol will aid in promoting what we call fluidity of the cell membrane. So it is the number one factor for also promoting fluidity of the cell membrane. Like we saw, the fatty acid does it to promote fluidity of the cell membrane. But what happens with cholesterol in promoting fluidity of the cell membrane? When cholesterol is high, when we have high cholesterol levels, cholesterol is too much, what will happen with the fluidity? Fluidity will increase. We'll also have high fluidity, increased fluidity. Why will the fluidity increase? Remember, cholesterol is a fatty acid. So if we have this fatty acid and temperature is when cholesterol is high, the fluidity will decrease, reduced fluidity. So this is what will happen with our cholesterol. If cholesterol is high, fluidity will reduce. Why? Because we are having, if this is our cell membrane, this is our fatty acid. By the way, cholesterol is always found within the fatty acids, here inside the fatty acid. So if cholesterol is high, fluidity will reduce. Why? Because we have cholesterol compacted within our cell membrane, within the fatty acid tails of our cell membrane. So high cholesterol, reduced fluidity. Then reduced cholesterol will have high increased fluidity, because now the cholesterol is further apart and we can have the fluidity of the membrane. But why is fluidity of the membrane also important? Because why fluidity is important, we can think of processes like endocytosis, and exocytosis. When we have endocytosis and exocytosis, the membrane will pinch. If this is our cell membrane, it will form a vesicle that pinches off, but the membrane also has to seal, to self-seal, to seal off that gap that was created. And in order for it to seal up the gap that was created, we need the membrane to be fluid such that it is not fixed, to be fluid such that it can be replaced again. The lost membrane can be replaced. That is sealing off this membrane. So that is how important cholesterol is. So these are the membrane lipids, the phospholipids, the fatty acids, and the cholesterol. Phospholipids, we said, are negatively charged, and they are always found in the exterior. The phospholipid heads, the phosphates are negatively charged, so they are found at the exterior or interior of the cell. They face towards the aqueous solution. Fatty acids, we say they are non-polar, and they allow non-polar molecules to pass through the membrane. Then we say they are responsible for fluidity, because if we have unsaturated fatty acids, they will form a kink or a bend that helps to improve the fluidity. Because when they kink, there is a gap created within the phospholipid. Then the cholesterol finally will help in maintaining fluidity of the membrane. But again, this cholesterol can work as a buffer. Before it helps in fluidity, maintaining fluidity of the membrane, but again it can work as a buffer towards temperature change. So it buffers changes in temperature, because if we have the temperatures too high or too low, if we have the temperatures too low, the membrane might freeze. But due to presence of cholesterol, it tries to work as a buffer in maintaining the temperature within the cell. But if it were not present, this cholesterol, maybe the cells would easily freeze, the cells would easily burn out. So that is how also cholesterol can be very important. But again, cholesterol levels are affected by things like temperature. If we have, let's say we are having temperature, a high temperature, if the temperature is increased, this increased temperature can melt the cholesterol. Can melt the cholesterol. So temperature can also affect the cholesterol. It can affect cholesterol. When it is low, also the cholesterol will come together. Then when it is high, the cholesterol will spread. So that is it for the membrane lipids. Then I think we can have a look at the glycocalyx. Look at the glycocalyx. So that is it with the membrane proteins, membrane lipids. Glycocalyx. So we are going to have a look at our glycocalyx. And we say the glycocalyx is a combination of two things. Those are the glycoproteins, and the glycolipids. So we say glycoproteins, we have a protein with a sugar attached. Glycolipid, we shall have a lipid. Let's say this is our lipid and a sugar attached to that lipid. So what is the importance of the glycocalyx? So the glycocalyx will help cells in cell identification. That is identification, that is self, what we call self from non-self. So identifying cells, let's say self from non-self. What do I mean? Let's say we are having cell A, and it has that specific region of the glycocalyx. Then we have also cell B that has that region of the glycocalyx. So if this one has a region of the glycocalyx, and this one also has a region of the glycocalyx. Let's talk about MHC1 let's say this is major histocompatibility 1. This one is used to identify cells that are like cells, MHC1. So if we have MHC1, major histocompatibility 1, and this cell interacts with this one will identify that it is the same cell. They are like cells. If your cells fail to do this, to identify each other, it can develop into autoimmunity. Autoimmunity, where we have cells, the same very cells in your body attacking themselves. So distinction of self, distinguishing self from non-self is that is one of the basic roles of the glycoproteins within our cells. Then another importance of the glycoproteins, due to the fact that they are always, if this is a cell, the fact that they are always spread along the whole cell, they limit water entering and moving out of the cell, so they can help to prevent dehydration or cells losing water, dehydration within the cells. Then again, these glycoproteins, another thing that can come into our minds is that we can use them in blood typing, can apply in blood typing, because the red blood cell will have specific regions of these glycoproteins carrying the, let's say this one is blood group A, let's say another one can be for blood group B, blood group C. So in blood typing, we can use the same criteria is applied of the glycoproteins, because if another person brings in blood with different blood groups, the cells will recognize whether that blood is self or non-self. If it is non-self incompatible, that is when you will have things like agglutination happening within someone's blood. So that is the basic importance of the glycoproteins. They are distinguishing self from non-self. We can apply this in blood typing. We can see it in the major histocompatibility 1, where we have MHC1, where we have self cells identifying the cells of the same type. Then they can help in preventing dehydration within the cells. So that is it with the second component of the cell membrane, which is the glycocalyx. Then finally, let us look at the third component of the cell membrane. And those are the membrane proteins. We are going to look at the membrane proteins. Membrane proteins. And we said membrane proteins, we have proteins that span along the entire membrane, but we can also have proteins within just the membrane. They are freely floating within the membrane. Let's say that is the protein there, but not spanning through the entire membrane. So we said we can have what we call integral proteins. The ones that span along the entire membrane, or intrinsic. An example is a transmembrane. They are transmembrane proteins. Then we also have what we call the extrinsic. So this one will be our extrinsic protein. The extrinsic proteins or peripheral proteins. One major difference between these two is that the integral protein spans along the entire membrane, the entire cell membrane. Extrinsic, they do not span through the entire membrane. Then again, integral proteins, they are high. They are more within the cell membrane. That is, I think, taking about 70%. Okay, they take around 70% of the membrane as compared to them. They are high as compared to the extrinsic or the peripheral proteins. And they commonly serve as transport proteins and pump proteins. So we need to know that their membrane proteins are either integral or extrinsic or peripheral proteins. Integral, intrinsic, transmembrane, then we have also extrinsic or peripheral proteins. We can call them extrinsic, peripheral. So that is it with the membrane protein. But again, we need to know generally what are the function of the proteins within this entire membrane. The function of the proteins. Number one function is that they can serve as enzymes. We need enzymes within the cell membrane. So some proteins can serve as enzymes within our membrane. Then we need transport proteins. So some can work as transport proteins. Here we can have the pumps, the protein pumps. We can have the channels. We have the carriers. All these are proteins aiding in transportation of materials in and out of the cell. Then we can have proteins serving as receptors. Receptors like for hormone receptors. We have hormone receptors. They can serve as receptors. Then we can have proteins aiding in what we call structural support and structural integrity of the cell membrane. So the integrity of the membrane can be maintained, that structure can be maintained by the presence of these proteins that span along the entire membrane. Then proteins can be used in communication between cells, among the cells, communication among cells. We can have proteins serving a role in communication. Then what else can these proteins do? Structure support. They can serve as receptors, transport, enzymes, communication. If we are having attachment of the cytoskeleton, it will take place on these proteins. So cytoskeleton attaches on these proteins. Remember, cytoskeleton, we say, is where we have the microtubules, microfilaments, and the intermediate filaments. So this is basically the structure of our cell membrane. So that is basically the structure of our cell membrane. So the main components, we said, we have the membrane proteins, the membrane proteins, the membrane lipids, and our glycocalyx. Proteins, we've seen a major division of the proteins as extrinsic and intrinsic. We've seen the functions of different proteins. The lipids, we said we can have cholesterol, the fatty acids, and the phospholipids, also serving different functions within the membrane. Our glycocalyx divided into two, the glycoproteins and the glycolipids. So another important thing is for us to know the differences between the cell membrane, and the nuclear membrane. This is very important, and the nuclear membrane. So differences between the cell membrane and our nuclear membrane. These ones are very important for us to cover. So a cell membrane, if we have a look at our cell membrane, the cell membrane is a phospholipid bilayer, but it's a single phospholipid bilayer. But the nuclear membrane is a double phospholipid bilayer. Double phospholipid bilayer. So for the cell membrane, we have a membrane like this. So it's a phospholipid bilayer 5. But for the nuclear membrane, we have a double. So we have a membrane which is, so we have this one, and again another membrane which is also. So it's a double phospholipid bilayer, but the cell membrane is a single phospholipid bilayer. So that is one difference between the two. Then the cell membrane doesn't have, okay, it's a continuous membrane, continuous without pores. But our nuclear membrane possesses pores. So this one has what we call the nuclear pores, the ones we saw in our video of the nuclear membrane. So if you missed out on the video, you can check out the structure of the nuclear membrane. So this one possesses pores, this one is a continuous membrane without pores. But again, the cell membrane encloses the protoplasm. Encloses the protoplasm. So this one is enclosing the protoplasm. The protoplasm, combination of cytoplasm and the nucleus, is enclosing it. The nuclear membrane is this one just encloses the nucleoplasm. Nucleoplasm is enclosed by this membrane. Then another difference between the cell membrane and the nuclear membrane, the nuclear membrane is let me start with the nuclear membrane. So this one disappears. It disappears or disintegrates during prometaphase of cell division, and reappears during telophase. This one is present throughout the life of the cell. Present throughout the survival of the cell. So this one is always present, but this one disintegrates during cell division. We very well know that this one will disappear. Then the cell membrane is also found in both prokaryotes and eukaryotes. So for this one, we know that a plant cell will have the cell membrane. Bacterial cell will have the cell membrane. Animal cell will have the cell membrane. But the nuclear membrane is only and only present in eukaryotes. So this one is only present in the eukaryotes. So eukaryotes, and this is what distinguishes eukaryotic cells from the prokaryotic cells. So only eukaryotes are possessing this, but this one is possessed by both prokaryotes and eukaryotes. So those are some of the differences between the cell membrane and the nuclear membrane. Then we can have similarities in these two, between the cell membrane and the nuclear membrane. We know that both are needed for survival of the cell. Both are needed or are required for cell survival. Both are important for survival of the cell. Both are contain phospholipids. Both contain the phospholipids, but one is a phospholipid bilayer, which is single, another is phospholipid bilayer, which is a double phospholipid bilayer. So both contain phospholipids within their membranes. Both contain proteins. Both have proteins for structure support. Okay, both membranes contain the proteins within them.